Thank you. Um, Thank you, everyone, for having me here. It really has been an honor and a privilege to be here to meet all of you in person. It's been a wonderful experience. I know it's only been a day, but it's been great to meet everyone and hear everyone's sort of stories. Um, and so I appreciate you giving me the opportunity to share with you some of the work that I've been doing. Okay, is is the system? But I'll speak louder. I can manage that. Um, so um, so what I'm going to talk to you today about is some information that you may already be familiar with, which was in that paper, and some of the newer findings. It's not complete. We're still doing the final data analysis, but for those of you who participated in the study when I was at UCLA with Dr. Martinez, I want you to sort of know what it was that you were contributing to and that it wasn't for naught. Um, let's see how we can advance this. So just to give you a little bit of background on my story, how I became involved in SKS. In 2016, I started my child and adolescent psychiatry fellowship at UCLA. I had always had an interest in genetics, sort of neuropsychiatric genetics, and had been looking for ways to train in that area. So all of my training up until that point had been in psychiatry and child psychiatry, but I was looking to figure out how to get more formal training in genetics. So I knew Dr. Martinez at UCLA. He's a medical geneticist, and had talked to him about maybe working with him and learning how to do research in rare diseases with him. For those of you who may be familiar with his work or not, this is some of the work he's done. He's really focused on overgrowth disorders. And the mTOR pathway for a long time. This has been his expertise. And so when I started working with him, he had a large clinic of kids who had disorders, within this area. And I met two kids with variants, mutations in the mTOR gene, but at that time it was still very, very new and very little had been studied, very little was known. And so after talking with Dr. Martinez about continuing to work with him and do research, we decided together that this would be an appropriate and good area for me to do my research in. And so I started working and talking with the families, and we sort of went from there. I was fortunate enough, once I formally started my postdoctoral training in genetics in 2018, to get a small pilot grant from the American Academy of Child and Adolescent Psychiatry. So that study was only possible because of that support and funding. And then I also started to collaborate with a biostatistician, Dr. Gerhard Hellman, who left UCLA and now in the University of Alabama, but he also helped with a lot of the analyses. So I just want to make sure to give everyone the credit that they deserve. So this effort is really between the three of us. And so the aim of the study that I initiated at that time was to better characterize the neurobehavioral profile of people, of mostly kids with Smith-Kingsmore syndrome. Being a child and adolescent psychiatrist, my interest was really in neurobehavior. And the hope was that we could characterize a very specific profile, and with the goal and intention of using this information for future clinical trials to try to better identify endpoints, right? Where are the strengths? Where are the deficits? What should we be targeting if we're going to develop treatments down the road? So the initial idea was to do local recruitment and have people come to UCLA to do a full medical assessment, neurobehavioral assessment in person, EEG, Gates assessment, and really the full shebang. A few issues came up with that. One was funding to get people to UCLA. It's not trivial. And number two was COVID. So that quickly became sort of not so feasible. So we sort of shifted our approach and wanted to be much more inclusive. Also, as more families became identified, it was all over the world. It was clear that this was sort of going to be global, and it wasn't practical to have people come to Los Angeles. have everyone come to Los Angeles. So we shifted it so that we could mail packets of questionnaires so that people could still participate in sort of a less sort of robust way, but nonetheless provide information about their children. So that was really, so the vast majority of patients who provided information in the study provided it through this sort of mailed packets and questionnaires. And then we also had people mail us their sort of medical records and imaging results so we could gather as much as possible. So in the end, what this was about the end of, around the beginning of 2019 or so, into 2020, we ended up with 17 patients, 13 boys, 4 girls. The age range was between 3 and 19, but the vast majority were on the younger side, the mean age of 7. This is the racial breakdown, the majority being sort of European descent, some Hispanic descent, and then Asian and mixed race descent. These are the countries where the patients came from. Again, the majority in the US, but also from the rest of the world. So we have global representation. This was the phenotype, the neurodevelopmental diagnoses, and the numbers at the end of the bars what had been previously reported in the literature at that time. So comparing what we have versus what had been reported. And the discrepancies, I think, are really just an artifact of the low sample size and sort of what was known to date. So about 40% of the patients had seizures, but again, the kids were very young. I think the kids in the literature were older, so more had sort of manifested seizure disorders by that time. Developmental delay, I think, was pretty similar. There was no formal statistic in the literature at that time, so it wasn't clear what we were comparing to. Again, intellectual disability can only be diagnosed by definition after the age of 5, and so I think that's sort of accounting for some of the differences. And then autism was pretty similar. We had about 40% of the kids had an autism diagnosis versus in the literature at that time, 30%. And I would take these numbers with a grain of salt, right? These diagnoses change as kids grow and develop. This is just what it's like think of it sort of like as a capture, like a moment in time, sort of a screenshot, if you will, when these kids were assessed. So this is a picture of the mTOR protein, if you sort of unraveled it and made it into a linear string, right? And these are all of those colored blocks are different functional regions, what we call domains of the protein, and the little numbers are just sort of an address along the protein, so we can track the protein's made up of individual amino acids, and we can track those locations. So out of the 17 kids who were included in the study, and this was in the paper that was already published, right? 7 of them had variants that had not been reported in the literature before. Some were what we call recurrent, right? More than one kid. The change at the protein, the amino acid 2406, right? We actually had 3 kids with that, and then you can see the other ones. Some are only in a single child, and some are in multiple kids. When I mean recurrent, these are these had been previously reported, and those were the kids included in the study who had those known variants. So this is sort of an overview of the genetic changes in the mTOR gene and protein that for the kids who were within our study. And so the idea was, well, first, and then just this is the additional phenotypic information we collected, right? When gathering all the medical history and information for the kids, these were some things that at that time had not been reported. Again, all of you are living with this, and so none of this may come as a surprise, but these were sort of new things that we identified that had not been formally reported in kids previously. And then, like I was saying, the main focus was this neurobehavioral assessment, right? We really wanted to understand what the range of neurobehavioral presentations were for these kids. So what we did was Dr. Martinez was part of a consortium studying a related gene, a related syndrome known as PTEN-associated disorders. And so they had developed a neurobehavioral battery for their kids over a long period of time with a lot of thought. And so instead of reinventing the wheel, we basically adapted that neurobehavioral battery with the real goal and intention of also being able to do what we call cross disorder comparisons. So once we understand what the neurobehavioral profile is for kids with SKS, can we compare it to related disorders and see what are the similarities and differences? So that was really the rationale behind this. And so this was the battery, the child behavior checklist that looks at emotion and behavior problems, the SRS, which looks at social impairments, being mindful that this is not a diagnosis for autism, because you can have other reasons why you might have social deficits, but it is a broad social responsiveness scale. The Vineland you might have heard of, and this assesses communication, daily living skills, and some aspects of social behavior. The Brief, which assesses the ability to manage thoughts, actions, and emotions, which is known as executive functioning. The SSP, which is an assessment that really looks at sensory profile of kids, so touch, sight, and sound, how they respond to those sorts of stimuli, and then something known as the developmental coordination disorder questionnaire, DCDQ, which looks at motor coordination, both fine and gross motor coordination, and then the RBSR, which looks at restrictive and repetitive behaviors, which are typical of autism. And so this is a very broad neurobehavioral battery with the goal of getting a broad general sense of neurobehavioral profile of kids. So this is just a very brief, targeted overview of what we found. We still have to complete the full analysis, and so I apologize if this is sort of anticlimactic, but this will be published more broadly, hopefully by the end of the year. But I wanted to give you a little taste of what we're doing, right? So the real goal is to do this genotype-phenotype analysis, right? Look at kids with the different types of variants, and look at their specific behavioral profile, and see if there are differences, but the groups become very, very small, right? So that is what part of the thing that makes it challenging, is to try to really identify meaningful differences when there's so few kids in each basket. So the only groups that we had enough kids to break it out into separate groups are the 1799 and 1977 variant. And you can see, right, this is just, for example, sensory functioning, where we have taste and smell, sensation seeking, and energy level. And you can see between the groups that there's a little bit of variation. And I don't think we can make any more sort of sweeping conclusions at this point, right? And this is the developmental coordination, right? Again, the 3 different groups, and you can see that it's all pretty similar. There's not major differences between the kids with different variants. Here, right, this is restrictive and repetitive behaviors, right? You can see that there's maybe a little bit of variation between the different groups depending on what variant the kids have, and then all the other variants. This is adaptive functioning. Again, you can see that there's some differences, but we need to more objectively quantify and do statistical tests to determine whether these are statistically significant, meaning is it just sort of random chance that there's differences, or does it really seem to be a meaningful difference? So I'll be done. So where are we going with this, right? We're really trying to describe in more detail this genotype-phenotype correlation in a more sort of rigorous, regimented way. We are also currently working with this P10 consortium. We have their data. We're doing cross-disorder comparison, so we'll have an overall profile of SKS and P10 cohort, and we're going to be comparing them to also sort of typically developing control group of kids with the same age, so you'll be able to see how SKS kids compare to another related syndrome, and then also typically developing kids. The take-home points is the last slide, is that we identified 4 new variants in 7 patients, some additional new clinical features, and we're finalizing the neurobehavioral profile for SKS. And this is my very big thank you to all of you, all of my colleagues at UCLA, and then the Developmental Synaptopathy Consortium. Those are the folks who provided us with the P10 data and the funding that has made this possible. So thank you very much. And
thank you. Thank you so much. Questions?
Not really a question, but for those that sometimes the science gets a little overwhelming, I just wanted to clarify genotype and phenotype. So could you give an explanation to the group what you mean by that?
About that, genotype refers to the specific genetic change that is associated with SKS, for example, the change in the mTOR gene that results in SKS. Phenotype refers very broadly to physical features or neurobehavioral features, clinical features of a disorder. And what genotype-phenotype correlation means is, can you identify specific genetic changes that are strongly associated with very specific clinical features? Is there a tight link between specific genetic changes and specific clinical features? So that's what we're looking for. And in my case, the specific phenotypic features is neurobehavioral features that we're looking for. That's the phenotype.
Thank you. Questions? I actually have a question. Oh.
This is actually more of a technical question about the histograms. The y-axis was measuring, what was the scale? I couldn't
Right. So
tell.
they're sort of arbitrary scales.
Okay.
Each one of those neurobehavioral tests has their own scales. And I didn't want to get and they get transformed depending on what sort of analyses you do. So it's sort of an arbitrary measure. And so I didn't want to, so sometimes the higher the score means sort of the more deficits kids have in that measurement. Sometimes higher scores mean the opposite. So I didn't want to sort of this is really short, so I didn't want to go into too much detail about these sort of arbitrary scales and all the transformations. But yes, it's a very good question, and I can understand if that's confusing. I wanted to just basically, the goal was to show you that we have the data. There are differences between the groups that are smaller rather than larger, and we will sort of quantify that more specifically once this is published.
So that was actually my question too, which is, I mean, we all know with our kids, it's when you see the registry, you can like tick, 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 tick. My kid has that, but the severity of it varies quite dramatically. And so what you're saying is that you're actually looking at the severity of it as part of your study?
Yes. So the assessments that we completed allow us to measure the severity of these specific neurobehavioral phenotypes, meaning repetitive behaviors, meaning sensory sensitivities, meaning executive functioning, meaning how well can they sort of execute on everyday sort of events.
Right.
So we have quantitative measures of all of those
Awesome.
things. The confusing part is the way that these assessments are designed. Sometimes a higher score means better, and sometimes a higher score means worse.
Yeah.
And so instead of sort of going through each one individually for the sake of time, I didn't.
It's perfect. That's great.
So I apologize for that.
Oh, no, it's awesome.
Yeah.
Any other questions?
Thank you for your presentation. Is this on? Okay. All right. So how are you working with the parents in the room in terms of all of us have paperwork that says, so all of us have paperwork that says what our diagnosis is, and then we So we all have paperwork that says where our mutation is for our loved one. So what is the go forward like tracking of all of this data in the room? How is that going to inform either a battery of tests, the phenotyping, et cetera? How can we help you, or how is the foundation helping inform the work, excellent, tremendous work you're doing? Maybe I don't. Maybe we already have that information.
So it's a very good question. That was one of the things we were talking about yesterday at the sort of research discussion, is we don't want this to all sort of go to waste. And we want to make sure that, one, the data that has already been collected is accessible to everyone in this room, in this community, moving forward. And number two, the idea so at the moment, there's no sort of longitudinal tracking, but I think that's on the radar for the SKS Foundation, where once this is defined, that in the future, similar assessments can be done for longitudinal tracking with the goal of informing these sort of endpoint measurements for trials.
It is complicated and 100% on our radar that the thing that our community needs is a very good organization of the variants we have, what behaviors we have, and how it changes over time. So we spent 2 hours talking about that yesterday. So this is most definitely on the list. Thank you, Dr. Besterman. He's going to be back up front this afternoon.